So my personal experience with ibrutinib in the relapsed refractory setting is that it really exceeded from the moment we were doing the clinical trial, all expectation. And I think it's best to relay it in terms of, patients' own stories. I had one patient who was on the trial, and because of his disease, he became anemic, he was very tired, and he just had to give up doing things around his house. And one of the things that he gave up was also shoveling snow. Now, why that would bring personal joy to somebody is beyond me, but it was something that he really lived for, to be able to, take care of his own home. And it was something that he wanted to get back to. So he started on the trial, and he was receiving ibrutinib. And when he came back to see us just a month later, his blood counts were so much better, his energy was back. He went back to shoveling his own snow. And to him, this was a personal accomplishment. And I think it's really important when we reflect on, activity of drugs. Sometimes we get lost in the translation of what drugs are doing. We get so trapped in response rates and progression-free survival that we really forget that we're transforming patients' lives. And this is really at the heart and soul of what we experienced when we were treating patients with ibrutinib. We were giving them new beginnings and changing their lives very dramatically. And I think what was really exciting in that trial was that even the number of prior lines of therapy didn't impact our responses. If the patient was refractory to their last therapy, which is usually a bad thing, they were benefiting just as much as somebody who had, relapsed. So this was a different drug than all the other drugs that we were using. And it spoke to the fact that we were targeting the essential mutation in Waldenstrom's, which is the MYD88 mutation. One of the things that was really exciting once we discovered this mutation was trying to figure out its signaling. And when we saw that BTK was at the heart and soul of how MYD88 was able to transactivate its growth and survival signaling, this is when we knew that we finally had the opportunity for a targeted approach to treating this disease. In commenting on the unmet needs going forward for patients with Waldenstrom's, I think BTK inhibitors and our ability to optimize their use in Waldenstrom's represents really an exciting challenge. We know because of the targeted nature of BTK inhibitors that these are going to become a very important mainstay of treatment for Waldenstrom's patients. And being able to optimize BTK inhibitors in patients that have the CXCR4 mutations is one of the challenges that we're currently working on. There are CXCR4 antagonists that are being developed, and we have a clinical trial right now looking at one of these in combination with ibrutinib in CXCR4 mutated patients. And I think we can expect to see more of these antagonists, become part of the mainstay of treating patients with Waldenstrom's. And it's important also to recognize that it's not just about BTK inhibitors, that there are other drugs that are also impacted by CXCR4 mutations. And so, being able to leverage this knowledge will probably end up aiding us in our ability to help patients, across the board with these mutations. I'm also very excited about venetoclax. This is another very important drug. We're seeing activity for this drug in patients that have seen prior ibrutinib and other BTK inhibitors. And we also know from a lot of the basic science work that we've done in our laboratory and has been done also in other laboratories that the combination of these two drugs can actually enhance activity of BTK inhibitor. And so for this reason, we're very excited about an upcoming trial combining venetoclax along with ibrutinib in patients with Waldenstrom's. One of the big challenges that still remains for us is understanding how to best treat patients who do not have a MYD88 mutation. This represents about 5% of all patients with Waldenstrom. And what we know now, because of the genomic landscape work that we just published, is that many of these mutations occur under BTK in the signaling cascades, and many of them are actually mutations that we see in aggressive lymphoma. So it gives us some idea of why these tend to be more aggressive presenting cases. And so being able to develop targeted therapies based on this knowledge is also an unmet need that we're currently working on.